pharmacology seminar, and we have Dr. William McEwan with us today. So William is a biologist by background. I have undertook a PhD at the University of Glasgow on filling retroviruses at their restriction by 3 proteins. He then undertook a postdoc with Dr. Theo James at the MRC in India, where he was a part of the team that discovered the intracellular antibody receptor TREM21. So William runs a research group at the UK Dementia Institute, where he also welcomed us here, welcomed us Sir Henry Dale Fellowship, and also a Lister Prize, researching how antiviral mechanisms can be deployed against aggregated proteins. So William, thanks a lot for your time in advance and those. Thank you. Thank you.
So how's the audio? Can you hear me okay? Yeah, great. So first, thank you so much for the invitation, Topic and Laura. Real honor to speak at Pharmacology. So yeah, I'm you at the UK Dementia Research Institute. And yeah, my background is in biology, and so interesting thing for me is that about 20% of organisms die every day from viral infection, and most of those are single cellular things. But it just underlies this very important war that's happening between hosts and the viruses that can kill them. And in terms of single cells, this really drives intrinsic immunity, the ability of single cells to recognize and destroy virus particles. And as multicellular organisms have arisen from single cellular organisms, they've retained that ability in their cytosols to recognize and destroy these parasites. And it's really, this intrinsic immunity is really sort of an underappreciated third arm of the immune system, as opposed to the more widely recognized adaptive and innate immune systems. So we're kind of applying this type of thinking to the problem of protein misfolding, particularly tau pathology, which occurs in Alzheimer's disease. So Alzheimer's disease is the progressive loss of neurons and the wasting of the brain, the enlargement of ventricles over time. And there are 2 protein lesions which were described by Alzheimer and Fisher at around the same time at the early part of the last century. And these are the extracellular beta-amyloid plaques and the intracellular neurofibrillary tangles, as they were called, which we now know to be comprised of protein tau. So in Alzheimer's disease, you get a serial progression of these different pathologies. The beta-amyloid, it's the first, which then stimulates the spread of the tau brain, which then results in larger structural trait changes, the loss of cognitive function. So the tau pathology itself, by electron microscopy, looks like this, and it has these 2 types of tau filaments, straight or paired helical filaments. And more recently, these have been solved by cryo-electron microscopy by Michel Goddard and Shaw Sherris at the LMB. And it's part of tau, this 306 to 378, which the tau protein is 440 and 441 actually amino acids long. So it's the small section of the tau that forms these amyloid cords. And so each of these is one molecule of tau, or region of it, which is stacking on top of each other to form these very, very long filaments, thousands of tau molecules long. And the reason we're really particularly interested in tau is that it is the strongest predictor of the cognitive decline. So the more tau you have in the cortex, the worse your cognitive scores. This is measured score where 30 is normal, very severe. There are low numbers. Moreover, if you have mutations in tau, there's about 50 of these now described. This drives the formation of these filaments and rare remedial forms of tauopathy. So this is not Alzheimer's disease. because there's no beta amyloid, which is a requirement to be diagnosed with Alzheimer's disease. So this is tau only pathology. And some of these mutations are very rare. There are just 1 or 2 families in the world, but they have this fully penetrant, dominantly inherited disease. And finally, the point to make is that most of us have some tau pathology if you're about 25. So tau pathology is staged in these Braque stages, with 6 being the most severe. That's being associated with severe Alzheimer's disease, all the way back to 0, where there's no tau pathology. By about 30, everybody has some evidence of tau pathology, and this progresses over time. So the older you have, the older the cohort you're looking at, the more of them are in these more severe tau cohorts. So the idea being, if we could target this tau pathology, we might be able to start addressing these diseases. So it's easy to say and very hard to do. So for the extracellular protein aggregates, such as beta amyloid, there are now methods by which these can be cleared, primarily monoclonal antibodies such as lecanemab, which combines the aggregates and immediately through innate immune pathways. For the intracellular protein aggregates, tau is one amongst many. There's TDP43 in motor neuron disease, alpha-synuclein in Parkinson's disease, as well as some rarer ones such as the fast clotwork. These aggregates are much harder to do, so much harder to address. So our idea really as a lab is, can we take these antiviral pathways and redirect protein aggregates? So gene we've been working on primarily is a gene called TRIM21. This is surprising discovery, which is that it's an intracellular antibody receptor. And that's surprising because it's the only one, as far as we know. Most antibody receptors are expressed at the cell surface. And the other surprising thing about it is it's an E3 ubiquitin ligase next to this N-terminal ring domain. So this C-terminus has a Price-Spry domain, the crystal structure of which was solved a while back by Leo James, whose lab I worked in, and some people who's in the audience. So the TRIM21 Price-Spry binds onto the antibody FC. So just to orientate yourself, this is the CH3 domain, and the fab arms would be sticking out here. All the other antibody receptors bind up in this hinge region, so up at the top of the FC portion, whereas TRIM21 binds this distal tip of the FC. And what we showed in a series of papers from 2010 to about 2014 is that TRIM21 acts as an antiviral receptor for viruses that import antibodies on their surface during infection. So most of these experiments we've done with adenovirus, and adenovirus enters through endocytosis. It then lyses the endosome and exists then as a virus particle in the cell. And if you supply antibodies against the virus during this infection process, these antibodies are also transmitted to the cell interior, where TRIM21 can recognize the FC and mediate the degradation of this entire complex at the protease. So this had some implications for virology. So this provides a final layer of defense against infection, and also changes where antibodies were thought to exert their neutralization effects. So before then, it was really thought that antibodies mediated their effect primarily as an entry blocking, so preventing uptake of virus particles into cells. So under this scheme, we kind of asked the question, could we start redeploying this process to cellular proteins? So if we could find some way of getting antibodies into cells, could we start targeting cellular proteins instead of virus particles? And a key breakthrough here was when we started working with Dean Cliff, who was a cell biologist who was working in oocytes. And oocytes are very easy for those people not to make, easy to micro-inject. So they can put molecules inside them. So we asked the question, could we introduce antibodies into these oocytes and test whether we could start targeting these intracellular proteins? So this is one of the first experiments that Dean did, where he expressed GFP and TRIM21 inside these oocytes. So the GF that merges up here, the GFP is green and the TRIM21 is in purple. So at the start of this experiment, he's injecting anti-GFP into the cells. What's going to happen? So what he observed is very quickly the GFP and antibody become associated, they coalesce into these bright dots, which then degrade very rapidly over the course of the cell. It's not very big. Okay, sorry about the distraction. So, second way of getting antibodies inside cells, what we knew it could principal work into sort of populations of mammalian cells was through electroporation. And we tried a lot of different electroporators, but this one is the neon, which worked particularly well. So we can introduce an antibody here against a cellular protein, IKK in this case, and observe its specific depletion over the course of a few hours. And here again, a second target. So this led to a kind of protein level depletion technique that we call trim away. So now coming back to tau, could we start thinking about addressing these proteins, this protein pathology that exists inside cells using this type of mechanism? And others in the field have shown that if you supply antibodies to mice models of tau pathology, you can reduce the amount of tau pathology in the brain. So here, with an active vaccination, immunize these mice against phospho-tau, and secondly, with monoclonal antibodies, where they're passively transferring an antibody against tau, reduce the amount of aggregated tau in the brain. The mechanism was really unclear, and I think this is gone into the clinic now. There's now several clinical trials ongoing with these tau antibodies, but really how they are supposed to be working, that's not being properly addressed. So the first idea is that they would prevent these tau assemblies, so preventing a kind of seeded aggregation response, so that a tau assembly that gets inside a cell can lead to the further propagation of that tau pathology. So you could prevent that process. The second is clearing through Fc gamma receptors, which the brain are largely expressed on microglia. And the third is promoting endolysosomal neutralization predation. And what we really asked is whether we could promote an intracellular response through TRIM21. So we showed last year that if we supply tau assemblies in the presence of antibody, much like we did with adenovirus, that these tau assemblies can bring the complex, can bring those antibodies inside the cell, they infect, as it were. And so here with an antibody called BR134, it's tightly associated with this tau assembly that we've supplied, and it becomes positive for ferritin. Ultimately, we found that the antibody didn't block uptake of the tau assemblies, so that the number of tau culture that were getting into the cell was unchanged by the presence of antibody. But the TRIM21 was recruited, so the number of TRIM21 positive tau content increased specifically when antibody was there. So following this, we went on to do the experiments in mice. This is a mouse model which expresses one of those mutant taus that causes early onset tauopathy. So it's called the PWS tau mouse. And we gave it weekly passive transfer of an antibody called AP42, which binds the C-terminus of tau. And what we looked at is the amount of insoluble tau in these mice, so that when we provided this antibody AP42, we saw a dramatic reduction in the amount of insoluble aggregated tau. But when we did the same experiments in mice that lacked TRIM21, this protection was no longer observed. So really suggesting that TRIM21 is a or the important mechanism by which immunotherapy is occurring. So there's now been 4 clinical trials of tau antibodies that failed against Alzheimer's disease, rare tauopathy called progressive supranuclear palsy. Very recently, just in the last few weeks, UCB have released data from their tau antibody called bepranemab, which is the first time reported reduced tau pathology in those patients. Unfortunately, there was no improvement to cognition. But I think the point stands that so far these antibodies have been designed specifically to prevent tau from being taken up into cells. And that's, from what we now know with TREM21, could be the wrong mechanism. Because first, by preventing the uptake of those tau assemblies into cells, you may prevent engagement of TREM21. And second, that those immune complexes are going to stay there. They're going to engage microglia, Fc gamma receptors on microglia, which is pro-inflammatory. That's probably something that you don't want to create or brain. And possibly consistent with this, there's now been reports of the first people who've had some of these antibodies. This is an antibody called losartanimab, which also targets tau. And this was this antibody was given to PSP cases, so progressive supranuclear palsy. And in the cases that were immunized, so this is the middle one and the right-hand one, they saw these very long, strange-looking microglia and astrogliosis, possibly consistent with an inflammatory response to the antibody. So based on all this, we're now undertaking some antibody engineering work to really try and generate antibodies that retain the efficacy through TREM21 and improve safety by dialing out the unwanted inflammatory interactions. So we're spending a lot of time trying to find the best region of tau to bind and then modulating the engagement of these various receptors to dial those properties in. And that's not something I'm going to talk about today, but that's really ongoing at the moment. So we asked the question with Leo James's group of how TREM21 becomes activated. And from various preliminary data, we thought that TREM21 was going to be a clustering activating receptor. So when it clusters, it becomes active, and when it's monomeric, it doesn't. So to test this, we designed a set of model substrates. This is GFP with tandem MIC tags attached to it. And against these MIC tags, we can use some of these very good MIC antibodies, one of which, called 9010, is rather unusual in that it binds in the clasping mechanism. So both Fab arms bind to the same epitope, which means that you get a one-to-one stoichiometry between the antibody and its epitope. So we mutated these MIC tags so that we had variable numbers of them that could be bound by the antibody. And we electroporated mRNA encoding these different DFP constructs into cells along with various antibodies, so to reduce trim away of the target. So this is a little bit confusing to look at, but when we put in a control antibody, we see no degradation. When we put in the 9010 anti-MIC antibody, we get good degradation when there are 4 binding sites available. If we start reducing the number of binding sites available to 3 or 2, we no longer get any degradation. So the control and the 9010 anti-MIC antibody look the same. And we can push this. So just looking at this target with 4 binding domains versus 3, that we see very good degradation or dose response degradation in response to the antibody for that target, but the one with 3 we don't see it. So this suggests a sort of threshold-based clustering activation, which we went on to show is caused by the dimerization of the ring domains at the TREM21 N-terminus, which is required for its ubiquitination activity. So this leads to a position where the TREM21 homodimer on its own is inactive. So its ring domain doesn't have a ubiquitination activity, but when it's dimerized, when it's multimerized rather, we can complement these ring domains together and start seeing ubiquitination and degradation. So based on these observations, we asked, could we engineer TRIM21-based degraders that are only active against aggregates, so that when they bind to monomeric soluble proteins, they don't get turned on. When they bind to the same target in an aggregated state, they will start degrading it. So we did this through two methods. The first is to sort of a minimal ring domain with a nanobody attached to it. And the second is what we call ring bait, where we fuse the ring domain to tau itself. So the idea here is that when this gets incorporated into a growing tau filament, the ring domains will be clustered and they cause the whole tau filament to become degraded. So this is the ring nanobody approach. So what we're looking at here is a field of cells expressing tau venus, aggregated tau venus. And we have a DOX-regulated ring nanobody degrader. So the tau in these cells is filamentous, shown by negative say in EM, but it's bound by anti-tau antibody and by an anti-GFP antibody, which detects the venus. So it starts this experiment, we induce expression of the degrader, and over the course of a few hours, we see these aggregates getting cleared. So this is this context where we have the antigen here, tau, in an aggregated state, and the activation of these ring nanobody degraders. And the ring alone, or the nanobody alone, don't have this effect. So then repeating this experiment in cells expressing soluble tau venus, in this context where we don't see any degradation. So confirming that this is a selective degradation technique. So we worked with David Kleinerman, chemistry department, to ask what the size of these aggregates are using super resolution microscopy. So at the end stage of this degradation, what's left over? And what we observed was very interesting, that the large aggregates were almost entirely removed. And smaller we got, that the worse the degradation, but there was degradation across the range of particle sizes. So this is then really a degrader with a very unusual property in that the larger and more aggregated its substrate is, the better the degradation, which is really the opposite of what the targeted protein degradation has seen so far, in that once you start getting aggregated proteins, they become very hard to degrade. So secondly, with the ring bait approach, so here we are using tau venus substrates. This is tau, so that we can see it, and then a ring tau degrader. So when we start expressing the ring tau, we start seeing the degradation of the tau aggregates. So compared to the ring nanobody, this is a much slower process, which I think if you think about it, it makes sense, because it's requiring the incorporation into the filament and then degradation. Probably a kind of cyclical process. How that exactly works, I'm not quite sure. But we can see the recruitment of the tau ring into these tau venus positive constructs before they would degrade. So to confirm that this is really due to the dimerization and the clustering, we made mutations at the dimerization interface of the ring domain, such as the M72E. So when you introduce this charge, it blows these two ring domains apart. And secondly, a catalytic mutation, the I18R, which prevents the ring domain from working. So introduction of either of these mutants or their combination substantially impaired the ability of the ring nanobody to degrade, suggesting that this is where aggregation is critical to that effectiveness. We also confirmed the pathway that we already knew existed for TRIM21, so ubiquitination, proteasome with the assistance of PCP, which is P97, which is an unfoldase. So inhibition of any of these three steps reduce the amount of degradation. So what I didn't mention in the introduction, but really is becoming important for tau pathology, is that the different diseases have different structures in them. So for Alzheimer's disease, you see these paired helical filaments, which I showed at the start. But in different other tauopathies, non-AD tauopathies, there are different tau structures. And the implications and the causes of this are not clear. So it could be that different tau structures are causing these different diseases, or the other way around, could be the different diseases are causing different tau structures. But nonetheless, this is now a really important area of study that address. So these are all wild-type tau structures, and in the field, we commonly use these mutants, the best that I've been using so far through this, which drive aggregations, they're pro-aggregant mutations, but they do not adopt these filament structures. In fact, we now know that they adopt completely different structures that have no resemblance to these great bright aggregates. So we were really keen to start trying to work with wild-type tau and develop assays where we could be much more confident that what we're seeing could be translatable to the human brain. So Melissa Huang, who was a PhD student with me, developed a wild-type seeded tau aggregation assay. So in these cells, she's expressing GFP tau or GFP tau with a progressive mutation and introducing tau seeds of various sorts. So as other people have shown before, the mutant tau versions of these lines respond very well to different sources of tau. So either from an Alzheimer's disease brain or these lab-generated heparin assemblies which can induce recombinant tau to aggregate with these, and also some AD filaments that she managed to propagate herself using cell-based assays. So these mutant assays respond very well to all these different types of seeds. Her wild-type tau assay was very specific. It responded well to AD tau and the AD tau that she made synthetically, but it responded very poorly to these lab-generated tau strains. So this now gives us much more confidence that what we're seeing is the assays are replicating something much closer to the disease-causing aggregates. So we use these types of cell lines expressing wild-type tau. Now asking whether seeds derived from the brain of an Alzheimer's disease patient or a progressive supranuclear palsy patient could similarly be targeted using the ring-based strategy. And so when we express the ring-based, these prevented seeded aggregate, that's a good control condition. And I think this is a very important property of this ring-based approach because we're using tau itself to direct the degradation. It can be incorporated into any configuration event. So to test these types of degraders in vivo, we started using these tail vein injections of AAV 9031, which crosses the blood-brain barrier. So when we introduce the virus, we get this very broad transduction of the mouse CNS, meaning that we start doing experiments that are very difficult. It's stereotactic injection. And if you're considering something like tauopathy, which is more or less global across the mouse brain at least, you need this kind of transduction if you're hoping to have any major effects. So in these conditions, we see tau pathology. This is the untreated or sham-treated mice. This is the tau pathology, and each dot here is a neuron full of aggregated tau. When we express the mutant degrader, which can't protect, we look protection. When we inject these ring-based degraders that are active, we see very good clearance of the tau in the mouse brain. So this is really the first time that it's been possible to selectively remove aggregated tau. So we could go on to ask questions of whether this leads to any phenotypic effects in the animals. So at 4 months of age, these mice are more or less fine. We're able to measure their gait using these mouse walker assays where each footprint can be picked up automatically and then turned into data. At 6 months, these animals have a motor phenotype, so they struggle to cross these perspex pathways. When we count these steps, that there's a number of steps required to cross. The time it takes to cross the walkway goes up substantially. So when we then treated one of these mice with a degrader, we found that it was restored to somewhat similar to the wild-type mouse. If you look, it does seem to have a little bit of stiffness in it, but it's certainly much better than the untreated condition. So quantified, that looks like this. So the number of four steps across with the ring bait didn't really change much between the 4 months time of treatment and 6 months when we ran out, whereas the inactive, the untreated condition suffered this most. So this really suggests that the selective removal of aggregates from the mouse brain is associated with behavioral improvements, which is very important. So really to conclude, what we've shown is that intrinsic immune pathways offer a route by which proteins can be cleared from the intracellular environment. And I think there's been long been an idea that these aggregates are intractable. There's nothing that really can be done to address them. I think this shows that if you provide the right signal in the right context and stimulate a response that is able. So in terms of immunotherapy, we showed that TREM21 is required for passive immunotherapy, which gives us a potential route for optimization of this activity, which we think creates a new type of antibody which could fight in terms of degraders, we found that ring nanobodies and ring baits can be incorporated or bind to existing aggregates and clear them. So that's distinct from immunotherapy, which we think only addresses the progression to cell spread of tau, whereas these intracellular degraders target that existing. And it means that we can get aggregate selective degradation rather than relying on other strategies such as ASOs, RNAi to knock down the genes. We avoid the loss of function that's inherently present in those RNA strategies. And I think the big problem for any of this is delivery. And there's AAVs are now being developed, which can get into the CNS. And there are some recent reports of unicured Huntington AAVs, which deliver the brain, having positive effects with a microRNA approach. So in terms of where next, antibodies are a possibility. It gives us hope that we might be able to improve them. They are, however, expensive. They poorly penetrate the brain. We have the AAV vector, which is also possible. Again, the rollout, it's probably very difficult given how expensive they are, and probably inappropriate for large-scale transduction. Where we really want to be is in small molecules. So reducing this activity down to a small molecule, PROTAC-like molecule, so something that will bind TREM21 and it gets the brain TREM21 up to a near substrate. Currently work within the stream. And so thank you very much. That's it.
William's very fascinating. Exciting. I must say the motor function just was really, really stunning.
Yes.
It's amazing. So any question from the audience?
Well, that was really exciting. I'm wondering about the original function of TREM21 with the dimerization that is necessary. So what is it looking for? What did it evolve for? The dimer.
So I think most virus particles or bacterial particles will have multiple antigens of the same copy. So it probably stimulates not a single antibody, because a single antibody could end up inside the cell erroneously, and you don't want to trigger a large response in response to that. So it's probably really just you're looking for a repetitive motif as is present in cells. So something that the adaptive immune system has already highlighted as important. Once that's identified, it will tell TREM21 to kill it. So it's is trying to discriminate between antibodies that randomly end up in the cytosol, which does occasionally occur, versus something that the immune system has already labeled as a threat. So that's the explanation we have to go into that. But that's why
it's very unlikely that something escapes endosomes with an antibody bound, that multiple antibodies nearby. Because with this low pH, for it to get into the cytosol, it has to go through the endosome.
Yeah, so I think it's a very low level of antibodies that get inside cells. So we have developed an assay based on high bit, large bit complementation, where we're tagging antibodies with high bit, and we can see there is a baseline level of antibody internalization. So I think it's probably picomolar or lower, but it does happen.
Any question from the audience? You first.
Very interesting talk. I want to know, is the TRIM21 still responsive in the aging brain?
It's a good question. It's definitely still there. I think we won't actually know until there's some kind of therapeutic trial, but the closest we've got is iPSC neurons. We've been showing that TRIM21 is active and present, and aged mice neurons, so these neurons that have tau pathology go very late. If the degrade is at a late stage of pathology, so 5.5 months, when there's abundant tau pathology, they're still capable of degrading at that late stage. So that's as close as it.
Regarding just the iPSC, how old is the iPSC for you?
The iPSC is always going to be a kind of fetal model. It's very hard to get an aged system. So I'd say the mouse is really the more relevant for testing the ways the proteotoxic stress from the aggregates, if not the age. So 6 months is not particularly old for a mouse, but they do have a lot of tau pathology at that stage.
Thank you.
Magic.
The experiments on Gator are fascinating. How long does it protect the mouse for? At what stage does the pathology start to come back again?
So it's hard to do that experiment because at end points, and once they start developing phenotypes, we can't keep them alive for that long. So that's quite hard to do. So once they do start developing the pathology after that, so we know that they do ultimately get the pathology. So it's a delay rather than a complete cure. But it is a very aggressive model. So how this translates, it's quite hard to sort of. But yes, I'd say it's a delay to symptoms rather than a prevention.
Is there any benefit in a almost prophylactic strategy? Probably getting rid of a nucleating event.
Probably yes. I think with the brain where neurons aren't readily replaced, the earlier you go, the better. I mean, just from that, prevention is better than cure idea. And I think the clinical trials are really pointing to that at the moment. So that anti-amyloid antibodies are suggesting that the people who don't yet have tau are getting better results than the people who already have established tau. So yeah.
It could be a tau-opty vaccine.
Yeah, I mean, it's an expense. I mean, as a monoclonal therapy, it's an expensive way. So I think you would probably need a good diagnostic, a predictive diagnostic, ideally, of who would benefit from that kind of.
Yes, please.
Hi, great talk, thank you. This is mostly for intracellular protein degradation. I want to ask you, so extracellular tau proteins exist. Is there a strategy can be used to degrade those proteins? Did you use endotag, which developed by David Baker group? Is that something possible?
Yeah, I think something like that could be a really good idea. I mean, I think that's actually been the strategy so far with antibodies, is to target the extracellular species. Unfortunately, with antibodies, even if you clear them, you do get an inflammatory response. So it might be that there's a benefit from clearing them and then a negative from the inflammatory response. So another way of doing that doesn't involve an inflammatory response, it's probably a good idea. It would prevent spread. It wouldn't address the existing receptor.
There is one question from the online audience. Is there a human phenotype of TRIM21 inefficient or null cell parts?
So there is rare mutations, heterozygous mutations in TRIM21 that are loss of function. So Leo James's lab showed this. And in vitro, those are non-functional. There is also, interestingly, a paper where they were looking for modifiers of frontal temporal dementia, and they found TRIM21 coming up in that. The relevance of that, I'm not quite sure. You wouldn't expect TRIM21 necessarily to come up unless there's an antibody response. Other people have reported antibody responses to tau and crosswalk aspects of tau. So it's not impossible that there's a little bit of antibody-mediated protection going on. But I'd say that's very much unconfirmed.
I have one question, Will. Is if you think about the small molecule approach at the molecular glue, does the system work the best when the target is an aggregated protein? So it's probably will be quite a bit challenging that if you want to bring the aggregated bunch with one end of a molecule and then the other one will interact with the trig. Am I missing something at all?
But you mean just the physical getting a trim next to
in the vicinity of the
yeah, it's definitely a challenge. So those I showed those filaments in reality in the cell and think they're kind of bundled together. So how that's actually going to look like and the molecule, it's probably a very difficult chemistry problem.
It's pretty great that at least
from the biologics we can. I mean, that's an antibody types. It just demonstrates it can be done. Just need to find chemistry that could do that.
Right. So any more question for anybody? Ritik.
Is there any other protein that binds a TRIM21 besides an antibody? So if you pull down TRIM21 from neuron cell extracts, is there something else that you get? Because I do feel they must have evolved for something else. It can't be you have a mutation with a phenotype. It can't be true. The only phenotype I would expect is an infectious phenotype.
So that's a very hard question to answer because there's a lot of reports in the literature of IPs against proteins of interest that pull down TRIM21. So there's a lot of, in the literature, interactors of TRIM21. How many of those are real interactors and how many of those TRIM21 binding to the antibody and then coming up in the identification of what was pulled down? It's very hard to answer. So if you look at the literature of what TRIM21 binds to, there are at least 100 proteins which have pulled down TRIM21 as an interaction partner. I think at some point somebody needs to do a sift and work out which of those is real and which of those is artifactual due to its so yeah, it's possible it has other functions. We do think its primary function is innate immunity. It's interferon regulated, so it's stimulated. When there's an infection, it's stimulated to produce more of it. It's at basal low levels, and then goes up a lot when interferon is present. So its immune function does seem to make.
And it's never released out. It's never secreted.
No, but there is a series of autoimmune diseases. So Sjogren's syndrome is one of them, where TRIM21 is a target of antibodies. Where that assembly is. So it's either TRIM21 getting out and interacting with antibodies, or it's antibodies getting in and interacting with TRIM21. We don't know which of those it is, but it's possible that TRIM21 gets released perhaps during cell lysis and then forms immune complexes outside cells. So yeah, we don't know, but quite possible.
You had one question.
You mentioned that there is potential for optimizing TRIM21 activity. So do we have already any molecules which act on TRIM21 associated pathways? And how do you imagine that mechanism of action?
So for the antibodies?
No, but
optimization.
I was wondering whether there could be any small molecules which act perhaps to increase TRIM21 expression or...
Right. So yes, as I said, it's interferon regulated, so you can give interferon, although that's probably not what you want to do. It's got a lot of toxic side effects. If there's something there's nothing specific at the moment. So I think what you'd really have to be doing is binding FIM21. So it's new binders that interact well with FIM21. It's what we're looking for.
Oh, it's a very stupid idea. When people do a vaccine, I think you have got a crippled sterile virus introduced to the body. Will that lead to some TRIM21, reservoir ready for next round of? I don't know. It's a weird way of thinking.
Yeah, I mean, it's interferon regulated, so a virus will upregulate it.
Okay, so great. So I think we probably can wrap up, and they join us downstairs in the breakout area to things like to give a big excellent talk. Thank you.
Great.
Started on what?
Right outside.
We have it.
Brilliant. Yeah. Okay. Thank goodness. Yeah. So you can take it. Yeah, we can already. I'm not chats with order about.
Great, that day.
Yeah. Yeah.
Something to kind of woman series in the ball that you look at the house. Yeah, it's like
But in the end, one is
Grace. Yeah.
I
mean, I'll talk to Laura. I don't.
Laurie. I think about what did you do? I'll show you. I'm quite on. I look at the bits and they just like to speak for it. And it's all smart, like they delegate.
Oh yeah. The ideas, which is why I keep reading. Is it where that they going? Also, they Cambridge. I didn't forget about and actually, I mean, we don't make any aggregate, but it's whatever.
So yeah, it's interesting. So there was a papers where they found FIM21 interacting directly with protein aggregates, and it is quite sticky, so it could have some intrinsic ability to deal with aggregates, and maybe that's why it came up in the frontotemporal dementia, and also functions. People have loss of function of FIM21. See that it's frontotemporal dementia with it. So it's not impossible that it has some intrinsic ability to.
There's people, I mean, the talk candidate was somewhere there for 21. I remove all the baggage because I expected them saw at the calories.
Yeah.
Like the antigens, and then realized in the 4 months, expecting like you wouldn't hurt. Okay, yeah, it's down to when they do, that'd be very flat. But you have all the reagents and we got really something interesting way. Yeah, back. Good, but please, yes, fantastic. And now we get it. Yeah.
Taking this. Thank you.
Yeah.
All right.